Welcome to the Introduction to Protein Synthesis lecture. Specifically, we're going to talk today about the differences between DNA and RNA. So if we want to review from our last unit, the structure of DNA is a double helix. The monomer of DNA is nucleotides, which consists of three parts: the deoxyribose sugar, the nitrogenous bases, and a phosphate group. You know that DNA is found in the nucleus, and the reason that the majority of DNA is found in the nucleus is because it's too big to leave the nucleus. We also know that DNA codes for genes that make proteins, and the problem when it comes to DNA is the genes that make the proteins are in the nucleus, right, because it's too big to leave, but the protein factory is in the ribosome, which is in the cytoplasm, not the nucleus. And so we end up with this issue of taking the code that is in the nucleus and try to get that code to the ribosome. And that is the process of protein synthesis that we're going to talk about in this unit. Since DNA can't leave the nucleus, then we're going to need a little helper, and that little helper is going to be RNA. What is RNA? RNA is ribonucleic acid. RNA acts as a messenger between DNA and ribosomes, and it carries out the process by which proteins are made from amino acids. And so we know that it's going to be that little messenger of the code from the DNA, because the DNA is stuck in the nucleus, and it's going to be able to take that code and travel that code to a ribosome in order to make the protein. What's interesting is there's a lot of different RNAs, which we're going to learn about today. So not just the one that's a messenger is an RNA, but the actual ribosome is an RNA, and the transfer RNA is an RNA. So we're going to talk about the different types. We know that protein synthesis is the process of making proteins from the DNA blueprint. And so here's our big picture, is we're going to have to take the code from DNA, because that's where it's found, and we're going to have to transmit that code into RNA, because that RNA can leave the nucleus, the DNA cannot. And then the RNA is going to use that code to create a protein. So what's the structure of RNA? RNA is a single-stranded chain of nucleotides. The thing that's different from RNA and DNA is that in that nucleotide, the sugar is a ribose sugar. It's not a deoxyribose sugar. So that's why we were trying to be specific in our last unit about the type of sugar in the nucleotide. It's going to be a ribose. And then RNA also has a different base. It has this base called uracil, and it does not have any thymines, and so it also has a different nitrogenous base in it. So you can see here, this is a single-stranded RNA sequence here. It still can form a helix form. Once again, the sugar is going to be ribose, and then also it's going to have a different base called uracil instead of thymine. So if it has a different base, well, then how the base pairs are going to act. So we know that apples go in the tree, A goes with T, G goes with C, the cars go in the garage. That's how we know the base pairing for DNA. For RNA, it's a little different. A is going to go with U, right, because we don't have T anymore. And then C is still going to go with G. And so another way to remember this is at the golf course is for DNA, and then at your golf course is for RNA. So just another way to remember those base pairs. Basically, uracil is going to replace thymine. And so if you still think ATCG, and you just know that uracil is different, is the base being used other than thymine, the base pairing here is still going to be the same, we just won't see that thymine. So given this DNA sequence, what would be the RNA sequence? Well, first we're going to have to find the complementary pair, right? So C is going to match with G, which we're going to see in the RNA, correct? Because G is present in RNA. A goes to T, but we don't have any T's in RNA, and so we're going to end up taking that T out, and it's going to be a uracil instead. T matches with A, G matches with C, A is going to match with T again, but once again, T is not a base in RNA, it's going to be a uracil instead. Then we have the C goes to G, T goes to A, and G goes to C. And so you're basically going to just find that complementary pair of the DNA, and instead of having thymines, you're going to replace them with uracils. And then you would have your RNA sequence here. It's just that complement to the DNA with uracil substituted over thymines. There are 3 types of RNA. We have mRNA, which is the messenger RNA. This is the RNA that's going to give the message from the nucleus, from the DNA, to the ribosome. There's transfer RNA, which is going to transfer the amino acids to make the protein. And then there's the rRNA, ribosomal RNA, which is the actual ribosome itself. So first, our mRNA, messenger RNA, what it does is it copies the code from DNA in the nucleus and takes that code to the ribosome. Once again, DNA is way too large to exit the nucleus to get through the pores, and so we have to translate that code into mRNA, and that mRNA is now free to leave the nucleus. The next one is transfer RNA, tRNA. This is going to transfer or carry one of the amino acids to the ribosome to match up with the mRNA to build the protein. So this is going to be the RNA that's going to physically add the amino acids together, matching with the other RNA sequence to build the physical protein. And then finally, our ribosomal RNA is the actual ribosome. So for the rest of class today, you're going to watch this DNA versus RNA video. And while you're watching that DNA versus RNA video, you're going to take some notes of the differences and similarities between DNA and RNA in the Venn diagram in your notes packet. After you watch that video, you're going to complete this table to tell me if it's part of DNA, part of RNA, or part of both. And so that's going to be your assignment for the rest of class.